Hey everyone and welcome to our new video. So in this video that we are gonna talk about polymerase chain reaction PCR, and also we will be talking about different types of PCR. So let's get started. PCR is a molecular technology aimed to amplify a single or few copies of DNA to thousands or million copies. So with this technique, the small amount of genetic material can be amplified. Also, if you are going to the history, it was developed in 1983 by Carey Mullis. However, PCR is now a very commonly used technique which are used in medical and biological research labs for a variety of applications. So let's talk very briefly on the setting up PCR reaction. So the constituents of PCR reaction is target DNA, pairs of primers, DNTPs, thermostable DNA polymerase, Mg2 plus ion and buffer solution. Apart from that, if you are talking about steps in the PCR reaction, mainly the PCR reaction is comprises of three major steps. The first one is denaturation, second step is annealing, and the third step is extension. However, here is the representation of the PCR reaction. The very step is denaturation, which means the double-stranded DNA has been unwound to a single-stranded DNA. This is the first step. And coming to the second step, which is annealing. So annealing is a step in which the primers are getting attached to the single-stranded DNA. That is what we call as annealing. And the third step is extension, which means with the help of DNA polymerase that helps in the extending of the DNA strands that would result in the formation of new DNA strands. However, let's talk on different types of PCR. So the first type of PCR is inverted PCR, which is also called as inside out PCR. This is mainly used to amplify an unknown DNA segment that flanks one end of the non-DNA sequence for which no primers are available. So the inverted PCR is commonly used for the amplification and identification of sequence flanking transposable elements. So coming to the second type of PCR, which is multiplex PCR. So multiplex polymerase chain reaction is mainly refers to the use of polymerase chain reaction to amplify several different DNA sequences simultaneously. And the third type of PCR is hot start PCR. So hot start PCR is a modified form of our conventional PCR, which reduces the presence of undesired product and the primer dimer formation, so due to a non-specific DNA amplification, so basically which increases the sensitivity and also the yield of desired target fragments. So coming to the fourth type of PCR is nested PCR. So nested PCR that usually involves two sequential amplification reaction. So each of which uses a different pairs of primer. So two sequence amplification reaction will occur, but in each of the reaction we will be using different pairs of primers. So coming to the next type of primer is in situ primer. So the low copy number of DNA can be detected with high sensitivity by this method. So it is widely used for the study of organogenesis and embryogenesis, and also it is used in infectious disease diagnosis such as HIV. So the quantification of DNA is also possible in real-time in situ PCR. However, the next type of PCR is long PCR. So the long range PCR is mainly refers to the amplification of DNA targets which are over 5 kb in length, which typically cannot be amplified using a routine PCR method or reagents. For a simple DNA template, long range PCR can amplify up to 30 kb and beyond, and for complex genomic templates, 20 kb is a typical target. So coming to the next type of PCR is colony PCR. So colony PCR is a method for rapidly screening colonies of yeast and bacteria that have grown up on a selective medium. So colony PCR is a very fast and a reliable method for the screening of recombinants. So coming to the next type of PCR, which is touchdown PCR. So in touchdown PCR, the annealing temperature during the first two cycles is set at approximately 3 degrees Celsius above the calculated annealing temperature. So the annealing temperature will get reduced by 1 degree Celsius for every 1 or 2 cycle. So coming to the next type of PCR is asymmetric PCR. So in asymmetric PCR, the end product is a single-stranded DNA as a result of unequal primer concentration. So the PCR is mainly carried out as usual, but with a great excess of primer for the strand targeted for amplification. So coming to next is bandstap PCR. So here, when the yield of the amplicon is very low, then the desired fragment required by gel electrophoresis and re-amplified with which is bandstap PCR. So each band of interest is sampled carefully with the help of a hydrodermic needle. So the DNA of that band is re-amplified. So coming to next is the anchored PCR. So a variation of the PCR technique that is applied to a double-stranded DNA fragment for which the sequence at only one end of the gene is known. However, the limitation of anchored PCR is it is relatively difficult protocol and a very large amount of starting template are required. Apart from that, it generates non-specific product. So coming to next type of PCR is reverse transcriptase PCR. So which is also called as RT-PCR, in which RNA template is first converted to cDNA using a reverse transcriptase enzyme. Then this cDNA is used as a template for the amplification using PCR. So coming to the next PCR is degenerate PCR. So degenerate PCR is very similar to the standard PCR, except the instead of using specific primers of a given sequence, here we are using either mixed primers are used in degenerate PCR. So that is the only one difference as compared with that of standard PCR. So coming to next is assembly PCR. So assembly PCR can be used to assemble two gene-sized pieces of DNA into one piece for easier for cloning or fusion gene or parts. So coming to next type of PCR is real-time PCR. So that is RT-qPCR as a quantitative PCR method for the determination of copy number of PCR templates such as DNA or cDNA in a PCR reaction. So basically we are using cyber green as most widely used double-stranded DNA dye. So the very next PCR is digital PCR, which is a kind of new approach to a nucleic acid detection and quantification that uses molecular counting basically. So that increases the precision by using more PCR replicates. So one of the advantages is no need to rely on references or standards. So that's about digital PCR. And coming to the next is methylation specific PCR, which is a method for the analysis of DNA methylation patterns in CpG islands. So MSP is a very rapid measure for accession of the methylation status in CpG islands. So coming to next type of PCR is ligation mediated PCR, which uses small DNA oligonucleotide linkers or adapters that are first ligated to the fragments of the target DNA. So this method is developed for basically DNA sequencing, genome walking and DNA footprinting. So coming to the very next PCR is overlap extension PCR, which is a variant of PCR that is also referred to as splicing by overlap extension or splicing by overhangs extension, SOE-PCR. So the very next type of PCR is VNTR PCR, which is a method targets area of the genome that exhibit length variation. So the analysis of genotype of the sample usually involves sizing of the amplification products by gel electrophoresis. So the next type of PCR is solid phase PCR. So this PCR is a very new method to amplify DNA as characterized by the use of surface bound primers. So this limits the amplification of 2D surface and therefore allow the easy parallelization of DNA amplification in a single system. So coming to the next type of PCR is quantitative PCR, which is qPCR. The amount of amplified product is linked to the fluorescence intensity using a fluorescent reporter molecule. So, however, coming to the next type of PCR is semi-quantitative PCR, in which the fluorescent data are collected after the amplification reaction has been completed. So, usually after 30 to 40 cycles, and this final fluorescence is used to calculate the amount of template present in the PCR. So, however, this method of quantification can give somewhat inconsistent results. So, that's all for today. Thanks for your watching and please do subscribe to my channel. Bye.